I am basically a neuropathologist, so I look this Alzheimer's disease as one of the model of pathology, and I will show you some of our results that we obtained in collaboration with various organizations. And we have collaboration with US Air Force Research Laboratory, Swedish Strategic Foundation. I am from Uppsala University, and also this support of European Office of Aerospace Research and Development in London and other universities. And as usual, I have to confess that whatever I am presenting today is my own view. It has nothing to do with any organization that I am working with. When I was a student earlier from an Indian university, I always think why people do get disease. Then we try to understand that we have a blood-brain barrier in our brain that is quite unique. And that might be the cause or that might be the result. I don't know. So we did this thesis and showed that stress induced by physiological points can open the barrier. At that time, it was not known whether opening of the barrier can lead to neuropathology. So now this part is much more prominent, and as you can see that when we try to study neuroprotection or brain pathology, we normally focus our information on neurons only, but the sense is that we have much more higher in number glial cells and even higher the endothelial cells. So that's why our research is focused on endothelial cells largely. And the good point is that we can still modulate by drugs these endothelial cells having receptors and modulate the function of blood-brain barrier. This is one of the examples of the blood-brain barrier because it is lying between the two tight junctions of the endothelial cells, and even it stops molecules. This is lanthanum, smaller than 12 angstrom. I did my training with Yngve Olsen in the University of Uppsala long ago. He's a neuropathologist, renowned neuropathologist. And then Dr. Servas Navarro from Germany, who is expert of electron microscopy. I owe my whole investigations to these two professors. And we have a hypothesis that any kind of CNS insult, including Alzheimer, opens this blood-brain barrier or blood-spinal cord barrier, then leads to different kinds of cascades causing cell death. And we believe that probably Alzheimer's disease is also not far away from these kinds of mechanisms. Well, why to try nanomedicine in Alzheimer's disease? I have just a fantastic presentation from the previous speaker who showed nanoparticles influence. And I mean, in this audience, these things are everybody knows, so I just skip it. The point is also that you can see psychologically brain is almost shrunk after the disease. And these are senile plaques. You all know, I am not going to show anything, and these are the position of amyloid beta peptide in the brain. These are pathological studies. And yesterday we had a very nice discussion on tau, its binding with microtubules. So, and these are the way they are formed. So, what is important? That in Alzheimer's disease, every component of the brain is influenced. Microglia, astrocytes, endothelial cells. But sadly, not so much attention is focused on the endothelial cells, the regulators of the brain, physiology, and pathology. There are a couple of previous publications. They are showing that blood-brain barrier is altered in Alzheimer's disease. And also there are some cases telling how cerebral microvessels are affected in situations of Alzheimer's disease. There are many enzymes present in the endothelial cells that regulate or control the blood-brain barrier function, and you can see that some of the enzymes are decreased or absent, some are increased. So these are the problems, that there are still great problems of neurovascular elements in Alzheimer's disease cases. Some of the, I sum for you, that endothelial cells are related and they are altered in Alzheimer's disease model. Then the question is that if we have Alzheimer's disease or any kind of insults to the brain, do we need one single item or single factor to correct this? I don't believe at all. because brain has so many factors that are released or altered in any kind of disease, which it is almost impossible to treat with one factor or one drug. So the point is that you know that brain itself produces different kinds of neurotrophic factors that can induce neuroregeneration. So I sum up some of the literatures where neurotrophic factors were employed for some aspect of treatment in Alzheimer's disease. Regarding nanotechnology, I'm not telling you the new story. There are previously some information already available as early as 2011 that nanotechnology could be beneficial in cases of Alzheimer's disease. However, this is a tool that can have both kind of effects. Neurotoxicology of nanoparticles is also very important before we consider as a good vehicle for the drug delivery. We used a drug called cerebrolizin. This is manufactured in Austria, and this is multimodal drug. It is a mixture of different kinds of neurotrophic factors from brain, glia, endothelial cells, and also contains active peptide fragments. This is commercially available, and there are also clinical trials going on in the United States in human cases of Alzheimer's disease. We did some experimental observations that how it can have some neuroprotective effects in different models of traumatic brain injury. First, we use transgenic mice, and if our hypothesis is correct, we can see this leakage of albumin in the cortical areas, and when we have given cerebrolizin in certain doses, we are able to prevent that leakage of albumin. It means that something is happening around the blood-brain barrier. Then, to have enhanced delivery of some of the drugs, we are trying nanowires. Although these nanowires were discovered for electronic purposes by Dr. Pudong Yang, we are using in biological aspect, and this is the titanium nanowires we are using to label drugs. This is Dr. Wang and this is Dr. Rang Tian. They have the US patent on these titanium nanowire, and they are the experts on labeling different kinds of drugs. Because of less of time, I'm not showing other aspects of these things. And I can tell you that even stem cells, when they are grown on nanowires, they can live longer, they can have better effects, and survive for longer time. So now people are using these nanowires in biology and medicine. And of course, these are very costly investigations, so we should have some government support. And since Dr. Rang Tian is in Arkansas, the former governor of Arkansas is helping us a lot. And we are very happy to get this National Innovation Summit top 15% prize to my wife here. She's sitting here on when we combine nano delivery of cerebrolizin together with nano delivery of alpha MSH in some forms of traumatic brain injury. So this was the innovation that was given a very high ranking technology at Washington, May 22 to 25. Previously, we also used this nanowire delivery for treatment of neuropathic pain, and in 2013, I was given 20%, not 15%, top technology award, and these things are. The point is that when we use this amyloid beta peptide immunoreactivity, you can see that nanowire cerebrolizin is the most effective than the cerebrolizin alone. You all know about amyloid beta deposition, and we measured biochemically in a mouse model, and you can see that nanowire cerebrolizin has the best effect in reducing this A-beta deposition. Same thing happened with tau protein, and you can see that nanowire cerebrolizin was the most effective in downregulating tau deposits in the brain. The point is that in normal situations, people have Alzheimer's disease and they are having many other kinds of other morbidity factors. For example, hypertension, diabetes, or even you know that brain injury is one of the important inducing factor of Alzheimer's disease, where our soldiers are significantly affected. This is well known. So what is happening if we have the combination of traumatic brain injury or hypertension or diabetes? I will show some examples today. So we have another model where these A-beta peptide is infused, and this is a standard model to produce certain aspects of AD pathology. And when we look at the cellular structure, the leakage of albumin, you can see here that this is the A-beta peptide deposition. Nanowire cerebrolysine reduced significantly, so as this albumin immunohistochemistry. Also, you can see that astrocyte activation was significantly reduced by nanowire cerebrolysine, so as the neuronal cell using Nissl staining. Then, since we are neuropathologists, we wanted to go to electron microscopy, and I can show you that electron microscopy, this myelin vesicillation in A-beta peptide infusion, and nanowire cerebrolysine is the most effective as compared with cerebrolysine alone. As I told you that diabetes is one of the inducing factor in Alzheimer's disease. So what is happening? I'm showing one example here in the case of diabetic animals made by streptozotocin. This albumin leakage and everything was so much increased and compared to the normal cases, and nanowire cerebrolysine reduced it, both neuronal, glial, and even albumin leakage. Then I will show you briefly some examples of traumatic head injury in the model of Alzheimer's disease. And we have this model of concussive head injury that is quite common in many aspects, including our soldiers. Of course, we cannot produce any human-like situation in animal model, because we must understand that we have to anesthetize the animals first and then apply head injury or whatsoever it is. While ever in normal situations, our soldiers or anyone can get injured when they are conscious. So we cannot replicate the model in animal situations, but we try to mimic those things. But you can see here that the injury was produced on the right side, but we have much more damage on the left side. This represents typical counter-coup mechanism in concussive head injury. So we think that our model is working nicely, and this is a table that is to shorten time. I'm showing you this. So you can see that these are the control groups, and we have this A-beta infusion, and this is traumatic injury plus A-beta infusion. Blood-brain barrier leakage is the highest. Edema formation also the highest. Neural injury, A-beta deposition, and also number of albumin cells. The point is that when we have given nanowire cerebrolysine, you can see that in traumatic injury plus A-beta infusion, they are the most significantly reduced at all levels. I talked about mesenchymal stem cells. We tried these experiments and also delivered cerebrolysine nanowire and also mesenchymal stem cells. And what you can see that neuronal damages were much more significantly inhibited by combination of nanowire cerebrolysine and nanowire mesenchymal stem cells, even after traumatic brain injury. Then, in the last few minutes, I will tell you one of the protein is neprilysin that is increased not only in Alzheimer's disease, but in Alzheimer's disease this is decreased. In brain trauma it is also decreased. So there is some hypothesis that if the drugs can enhance this neprilysin in the brain or CSF, we are going to have neuroprotection. We tested this hypothesis in our experimental model, and the point is that neprilysin is always associated with A-beta protein, but not so much association could be seen with astrocytes. Of course, to reduce the time, I'm showing this clumsy table, but you should focus here on this. Nanowire cerebrolysine plus A-beta infusion, and here is nanowire cerebrolysine plus nanowire neprilysin. What you could see that all these parameters, A-beta deposition, blood-brain barrier permeability, edema formation, and also this neuronal changes were maximally protected when the combination was given in the model. This is an example of hippocampal changes, and as you can see that cerebrolysine plus neprilysin treated, the hippocampal cells were most preserved as compared to the only untreated group after injury. Then we're talking about Dr. Sir Kroto, and he believes that we should try different models of nano delivery of the drugs, maybe the same or different, to see what happens. He is now in Britain. So at this point of the moment, we can only say that co-administration of nanowire cerebrolysine with other drugs is very effective. I will show you some of the examples. The point is that what will happen if we deliver the same drug using another method of nanodelivery? And I'm showing you one example here. We have used PLGA loaded cerebrolizin with our colleague Giovanni Tosi in Italy. And you can see that drug can be released. I can show you only one example here, that after the blood-brain barrier leakage in this model, TiO2 delivered cerebrolizin is much better than PLGA level cerebrolizin. Of course, both are significantly reduced, but if we can compare in between, we still feel that this titanium oxide nanowire induced cerebrolizin is much more effective. Why we have problems in animal cases and the human? We can't replicate those of the drug treatments in human cases because probably no two human cases are identical. Therefore, from lab to the clinic is very difficult to reach. And here we are talking with Dr. Shelley, who got this Nobel Prize in 1977. He has some CRF fragments. Now we are using nano delivery of CRF fragments in models of Alzheimer's disease. At the moment, I cannot disclose the results. It can be told later. And here, this is the lady, Dr. Asia. She is working in Fayetteville, and she is developing another kind of leveling, this titanate nanospheres when the cerebrolizin was leveled. So this is the model. We put cerebrolizin, she put cerebrolizin in these titanate microspheres, and you can see that the cerebrolizin release is quite prominent. And we have seen these results, and we can say that this is also working very good. So at the moment, I can only say that blood-brain barrier is damaged in Alzheimer's disease. Nanowire delivery of drugs is quite effective, and other factors, for example, even nanoparticles, diabetes, hypertension, and other comorbidity factors also affect pathophysiology. So we believe that future belongs to some aspect of nanomedicine, but we should not ignore the comorbidity factors that influences many types of neuropharmacology or even clinical trials. We are thankful to many other supporters here. I'm not going to read. These are different organizations that support our research. These are our collaborators. And I am from Uppsala University. Thank you very much for your time and attention.
Thank you, Dr. John. We have a question.
Hi there. I was just wanting to get a bit more detail on how the silicon nanowires work and how effective it is in delivering drugs across the blood-brain barrier.
We have used titanium oxide nanowires, and we are examining when the drugs were labeled with titanium nanowires and given either intravenously. We have analyzed these titanium fragments in the brain using electron microscopy, and we can see that these titanium nanowires can penetrate into the brain. And the point is that these titanium nanowires are innocuous in our hands up to 7 weeks of administration, so they are very good. The other way, we have also examined the drug titer in the brain, and we can see that when these drugs were delivered, either PLGA or titanium dioxide nanowires, their level is much more increased than they are given alone. So we believe that there is a facilitation of the drug. The other point is that they can last longer in the brain to have this long-lasting effects, because probably the drug metabolism is reduced due to their binding with nanoparticles, either PLGA or titanium oxide nanowire.
So the drug is actually attached to the nanoparticle and then presumably delivered intravenously?
They could be delivered intravenously, yes. And these leveling is done in the lab of Fayetteville, and they were characterized by different kinds of X-ray diffraction studies and all other things. So these techniques were very standardized.
And just finally, in terms of percentages, what sort of percentage increase in drug delivery might you see across the blood-brain barrier?
In our case, we are using traumatic brain injury in Alzheimer, where the blood-brain barrier is already broken down. So the drug level could reach any time. It depends on the time, of course, but probably 75 to 80% can reach to the brain.
Okay, thank you.
It would be very interesting to know whether the pathophysiological improvements go along with functional and cognitive improvements, of course, because there have been a lot of drug discovery work where people cleared amyloid load and plaque and so on, but the cognition didn't improve. So do you have any data on that at all?
Thank you very much for raising these data. Since I have only 20 minutes, I did not show them, but I can tell you that we have measured cognitive functions, some of the functions, for example, using rotarod, using inclined plane angle, and also finding a platform, a hidden platform, and we can see that in those situations when we have neuroprotection, they are also doing better on cognitive functions. But I do not have time, I did not show it. Yes, we have the data.
Right. Thank you very much. Thank you, Dr. Sharma. We appreciate that. And our final talk before the break is from Florida, US, and Dr. Shugaya.